All right, in this video, we're going to go over the general slides this week, but we're also going to talk about WA2. I know I put off doing this because I wanted to get it exactly right, and because this is a difficult thing to explain sometimes. And I know there are previous videos with chi-squared, things like that, but you got to know what to put in there. So I'm going to go slow. There are going to be lots of videos here. Have to do this and hopefully step you through this. But the first thing we have to do is go through these slides, and let me just discuss these slides. They'll be up on OneDrive here. Oh, probably this is Tuesday, so Tuesday night these will go up on the OneDrive. These videos will be available here Tuesday night as well. I know you don't have as much time, but it's better that I don't tell you wrong something, because or tell you in a confusing way. I want to make for sure that you get this, because it can be very confusing. We can save time for everybody just starting out on the same page and getting it correct. So let's start here on week 7. It says October 11th at midnight. Just understand that you know the due date. I don't know if you can talk Dr. O'Brien into changing that, maybe. But he knows that several of us are getting a slow start on making videos for you, because this is difficult. I'm going to also introduce WA3 a little bit, the molecular biology overview, pipetting, agarose gels, lots of stuff kind of to talk about here. But let's get this out of the way and we'll keep going here. WTM 1 and 2. It's kind of confusing with the WTs and WTMs, wild type or mutants, etc. But just keep in mind, we're try to keep everything straight. Here we go. This is your mating scheme. So this is what you want to put into the computer software and have it spit out some answers to you. And I'm going to suggest that you do this many times and record your data. So when you run this, you don't want to just run it once. Remember, there's some variability in the software. So it will give you numbers. Say if you do it with 100 flies or something, and you're getting you know, a couple of hundred of this type and a couple of hundred of that type, etc. There's nothing wrong with you doing this many times and adding all of those together as if it's one big population. And the reason you do that is because you get closer to the real answer if you do it. The way the software is designed, it's designed to kind of imitate the real world. And if you know anything about statistics, the larger sample size you have, the larger population you have, the closer you come to the real answer. So the more you run, the better answer you get. And so if you do more flies, if you're running it and you're not getting the answers maybe that you think, maybe you should use more or run it more times up front, and in other words, start out with a sample and cross. Instead of, you know, 10 flies, start with 100, or start with 500 for that matter, and make it run a larger sample size, or do it several times at least. And you will probably come out with an answer that's closer and less variable, all right? Because the software is supposed to imitate the real world, and in the real world you would have done things like, oh, you let a fly out and you killed one here if you were working in your lab, and your numbers are never perfect. And even then, even if you did it perfect, your numbers are never perfect. They vary a little bit. But statistically, you come up on average with the right answer. But if you do more crosses, you have more flies mating, you are more likely to get the right answer. So I just want to put that up front to tell you to cross more flies, and you'll actually save yourself some time maybe later on in case it came out a little off. So let's start up here, and what are you crossing? You're crossing here, what? A female, and it says 3 to 10 wild type. So if you're going to do, say, 10 and 10 here, these are males, and you're going to cross these two sets of flies. Now you have to know how to set this up. You have to know how to, in the software, to set up these particular mutations in the male, and you have wild type females. That's pretty easy to do, and you're going to cross these, and if you're going to do 10 and 10, well, don't just do 10 and 10. Try to do it 5 or 6 times or something, and add those up. That might help you out later on. I'm just trying to give you some. It might work perfect if you do 10 and 10 and you just keep the minimum number, but it's more likely to give you correct answers if you do more. That's all I'm going to say. Now you go through this and you're going to get your result, and that'll be your F1. Okay, these are your parental flies and these are your F1. So you also need to keep track, all right, of what's going on. So make sure you know who your F1 are and you have all your statistics. And so when I do this sort of thing, I use one whole sheet of handwritten paper and I put all my results here, and my F1, I put all of that on one piece of paper, and then on the back I do my chi-square. And then that piece of paper is separated and everything is right there. So you're going to get your results through the computer, and I recorded on handwritten paper first. I put it on the computer later, maybe in a spreadsheet. You can do this in spreadsheets, that sort of thing. I will make another video on chi-square working with this. I've given you one chi-squared program already, or video already. I will go ahead and tonight and make another. I'll probably make 3 or 4 videos tonight, just because I'm getting a late start. I need to give you as much information so you can get done, but I want to make for sure you get it right. I don't want, I would prefer to make some extra videos than 20 of you emailing me. I think you can understand that. So I'm going to try my best, but I also don't want to scare you off from emailing me, especially if you don't understand. I will help you. But let's go on to the next slide here. And I have done, if you want to ask about WA1, I've graded all of one class, but I haven't posted them. I've graded some of those grades. I think everybody who made an A, I put those out front. You worked all that. You worked really hard to get an A, so I thought you should get your grades first. So there were in one class, I'm not going to tell you which one, but in one class, let's see, the number of B's and C's are about equal, and then there's 1 D and 1 F. So I think we got 4 or 5 A's, and I believe there were about 8 and 8 B's and C's, and then 1 F and 1 D. The other class should be probably very similar. Don't know that for sure. I haven't finished the second class, the sections, I should say. So just if you're wondering how the grades are turning out, actually I was pleasantly surprised. Most of the time on these, it's more like we have 10 or 15% Fs, and as far as I can tell, we had 1 F and 1 D. That's pretty good. You guys are way ahead. Of course, this is a little slightly easier assignment than we normally have for this one. We'll try to get around to WA3. I'll at least talk about it. Probably be a video. It probably will not be. This is Tuesday night. That'll probably be Wednesday before I get to WA3. Let's see. Complementation. So let's talk about this. It occurs when 2 different alleles, usually at a different gene loci, encoding proteins of a different function together, each provides something the other lacks. So what we're talking about here is they enhance one another or alter one another in some way. So you have 2 genes both producing a protein, and they both have their own individual function, but one of them complements the other. So if you're wearing clothes, the way I think of this is you have 2 things that complement one another, or food. Sometimes you have you have 2 different foods, but they kind of go together. The food is better if you put them together. That makes sense to me. So it's a similar way. Not quite the same idea, but you can think of it that way. And in flies, 2 strains will have 2 homozygous recessive mutations that produce the same mutant phenotype. In other words, you have 2 flies, all right, that have a homozygous recessive mutation. So homozygous, that means like little a, homozygous recessive. And there are 2 different ones. So let's say little a, and little b. Now, they produce the same phenotype. This example is they change the wing structure. Well, I think an easier one would be like in a human, maybe you got something like they both cause you to have um, blonde hair. And when you cross these, all right, um, they have offspring that will produce wild type phenotype, normal wings, or in this case, maybe if instead of blonde hair you get black hair. All right, we'll assume black hair is human, and that's the wild type. It's not. There is no. It's more difficult with something like with hair color in humans, because it's about 12 different genes that give you the hair color you have. But you could understand how that might work. So the normal wings are produced when you cross them, and of course, why is that? That's because when you cross them, you have, yes, one fly has little a, the other fly has little b, but it also means they probably have big A with the little b, and you have big B with the little a. So you're going to end up crossing them, and you're going to get heterozygotes. And let's see if this explains it reasonably well. Yes. So we look up here, and this is actually from last week too. We kind of talked about this. So in other words, you have little b, and big A. Well, they're going to give you the exact same fly, and little a, and big B. You cross them, you're going to get the exact same flies. But if you cross these two, what do you get? You get a big A, little b, and a little a, big B. So ultimately, what you end up with is a big A, little b, and a big B, little b fly, and you end up with something very similar. You get a heterozygote in both at both loci, and with heterozygotes at both loci, the dominant gene, of course, is in charge. It's going to dominate over the small letter, and therefore you're going to get what looks like a normal fly, a wild type, right? You're going to get the dominant one, assuming the dominant one is the one that you find in the wild type. So this is an odd one, but it's recessive. It carries. You call this type of fly a carrier. It's a carrier for the little a and the little b allele, and but you're going to get it, and that's kind of what you're looking for here. You're going to want to record these, and this is called your complementation cross. All right, so keep in mind what we're talking about here. Now, complementation, you don't have to do it with wild types. Wild type's the easiest example of this, but here they're doing it with a black body color, and then they're going to end up with this fly that ends up looking normal. You can use this. You can use this in a lot of ways with a complementation cross. A complementation doesn't mean you have to start with the mutants, though they do here. But if you do, the idea is to make for sure that you have all normal or wild type looking flies. The phenotype looks normal in your F1. All right, and so you examine them, you count them, etc. Now, this would be what you're doing if you were in the lab, and this is easier to explain if people have actually been in the lab and been doing this all semester. And it's kind of hard for you guys because you don't have that tactile learning where when we talk about this, you've actually done it, you've counted the flies. This is all kind of virtual and hard to grasp sometimes. I actually suggested when we did this that we mail you the flies and you do them on your own maybe, but yeah, nobody thought that was a good idea. It would have been difficult. We'd had to make videos showing you how to do a lot of things, and you'd had to knock out the flies on your own using ether. It would have been a mess, but still, at least do it once, I thought, so that you could get an idea what it was like to cross them. But that would have been, I think you would have enjoyed it, but nah, I agree now that it's probably too much to do. Here is your WA3 rubric. Now here, this is how I will grade you on WA3. I'll go back and do WA2. I have that. Actually, I should probably do that. No, we'll do it at the end. I have this right here. your WA2 writing assignment. I'll do that in a different video. But I will upload this. Actually, I don't have available. I just have it in a slide, so I can't do the. Oops, what happened there? I can't give this quite to you yet. But the molecular genetics, the ultimate goal of all of this is to identify, using these classical techniques, these breeding techniques, to identify and prove that your mutant really is the one you think it is. So the original way, the classical type, is through phenotypes, right? And you cross them, and if you get what you think you get, you do the math, you do the Hardy-Weinberg, the chi-squared, you should get numbers that support your hypothesis. The molecular approach, which is what we're going to be going into soon, will be to actually identify the gene that causes it and then analyze it that way, and that's where we'll get to the bioinformatics. All right, and this is a different type of genetics. We've been doing what's called classical genetics. That involves breeding flies. We're about to move on to molecular genetics, and then within the molecular genetics, we're going to be doing bioinformatics, which is letting the computer essentially help you do it. So historically, we've gone from classical genetics, that's like Mendel, and that's what you typically learn in your typical genetics class. Then you go on and you learn more advanced genetics after you do molecular biology, and that's called molecular genetics. And then one step above that is the bioinformatics, which are the tools, the computer-based tools that let you analyze the molecular genetics, the data that you receive from doing your experiments in molecular. Now, the molecular part we will discuss, and we usually have labs for doing this, and we will continue doing that. But the molecular genetics part, we will do this virtually as well, and I'm about to explain some of the things we're going to do. You do need to know some terms here as we get started. There are point mutations, all right? Sometimes you'll hear the term single nucleotide polymorphisms, or SNPs, or they'll also call them SNPs, because SNP kind of, if you were to say it, would be SNP. But single nucleotide base changes, or single base change, it's called a point mutation. You should also be familiar with the term SNP or SNP, single nucleotide polymorphism. You should also be familiar with transposable elements. We are not doing transposable elements here. We've taken that out, but these are jumping genes. They are whole sections of the genome that will literally pop out, especially during that phase where you're doing the crossing over, and they'll jump to a whole new chromosome. All right, they don't have to, they could jump, but they'll jump to a new location, and it's just not the exchange of pieces like I've explained in a video, but they actually go to new places and pop right in the middle of a gene somewhere and cause all kinds of chaos. But this is actually what I study a great deal, is transposable elements, and probably we can probably trace back the evolution of humans from chimps to transposable elements. It very well may be that the majority of mutations, the majority of evolution is done by transposable elements and not by these other types, which, if that can be proven true, would kind of upend a lot of current thinking. But there is more and more evidence that these are way more important than we ever knew. These are not as important. Insertions or deletions are similar. They're smaller than a transposable element. These are just like usually one or two bases at a time, called an insertion. Of course, you can understand that. And here you're just changing one. You're changing a G to a C, or vice versa, something like that. Here you're adding an extra one or taking one out. And these are usually worse than these. These are the least damaging, you might say. These are more damaging depending on the size. These are usually these cause a lot of chaos, and but they may be the ones that are most important for the evolution of one species to another. All right. Now, what you do if you're doing molecular genetics, you extract DNA from your flies, and then you amplify it using something called PCR. And PCR is the polymerase chain reaction. And I have several pre-made videos on this. I may make a new one for you. But PCR allows you to take a small amount of DNA and make a lot of it. It's amplified. So they use the term amplify. But it just means take a little bit of DNA and make a whole lot of it. Because of the little bit, you can't do too many experiments. And you know you're going to run out of it. You need a lot more to do certain types of experiments. After all, DNA is small. You may not have a big sample. Maybe you just have a little bit of blood, or flies aren't big. How much can you get out of that? Well, you need to amplify. You need to make copies. And so all PCR really is, if you really want to think about what PCR is just replication outside of the cell. So if you've had biology and you've learned replication, transcription, and translation, well, replication outside of the cell, if you can manage to do that, you have PCR. And that's exactly what they did. They copied replication. They use artificial enzymes that do the same thing, that the DNA polymerase, which copies the DNA. They used it outside the cell, and basically they use it to just make many copies of existing DNA strands. Of course, they use certain things to only amplify the pieces you want, like between this base and this base. And you can get, most of the time PCR is only done with, simple PCR is only done with 500 or 600 base pair. You can get more than that, but generally they do it in little chunks like that. Of course, sequencing the entire genome, that's why it's difficult, because you usually have to amplify it. That means you have billions of bases and you're amplifying it 500 at a time. So they have special machines for this, and they take the laboratory work out of you. You just put the sample in the machine and the machine does it all. All right, I shouldn't say that. It's a lot more difficult than the way I just described it. All right, we're going to analyze our DNA sequence and identify the unknown gene. That's where we're getting into more of the bioinformatics. Micropipettes. Hopefully you've used those in other courses. Maybe we can make some videos showing you their correct function. I don't know. Normally we do that. This is a micropipette. Hopefully you may not have known what I was talking about when I said it, but maybe you recognize these from a class you've had. This is how you operate them. The names, press, the first stop and second stop. If we make a video, I'll explain those. This is how you would set the micropipettes. These are the types of things that you might be tested on, by the way, in a quiz from Dr. O'Brien. This is our gel apparatus, which pulls it, and I have lots of videos on how this works. I'll post those for you because those will be helpful to you so you understand how this works. This is the result. Now this is a gel, and I have videos on this as well, and I have a little exercise and I'll post it. You certainly don't have to turn it in, but certainly if you do the exercise I give you, you will certainly know how to answer the questions on it, and I'll give you the answers and everything. All right, so review writing assignment 3. Well, we haven't quite gotten there yet. I'm going to do that probably tomorrow, and I'll try to find some more of these older videos maybe and organize them for you on the YouTube site. All right, I will do this part. This is WA2. I'll do this in a different video.